The study of DNA evidence from blood or skin tissue left at a crime scene is a large part of forensic biology. DNA, or the molecule deoxyribonucleic acid, is found in every cell of every living organism. Obviously, our cells can only be seen under the magnification of a microscope. But when a cell is observed under a high-power microscope, it doesn't matter if it is a cell from your skin, blood, brain, heart, etc., you will be able to see chromosomes inside the cell. Chromosomes are tightly wound up and condensed strands of DNA around some proteins. Stretch out the chromosomes and you can see the DNA structure. DNA is made up of 4 connecting molecules called base pairs, sometimes abbreviated as A, T, G, and C. Even though every cell in your body contains your same DNA code, the arrangement and order of these base pairs is specific to each individual organism. In other words, the order of your DNA base pairs in each of your cells is unique only to you. Again, from the cells comes 46 chromosomes, or 23 pairs for humans. Half of your chromosomes came from your dad, and half of your chromosomes came from your mom. Biologists and forensic biologists study the chromosomes in several specific areas that can be observed when they put chromosomes through a banding technique, and in this case, a fluorescent dye process. So here is a section of DNA from one person. The different colored bars represent their unique order of those base pairs. This section of DNA might code for a trait. We call this section a gene, and it might be the code for our cell to make red hair color, or brown eye pigments, or to be nearsighted, or be able to roll your tongue. Of course, another person might have their own unique code or order of base pairs for this gene. Now here is where we can begin to learn about how forensic scientists use a DNA sample from a crime scene. Forensic biologists like to use the sections of DNA that don't code for a trait. They are the filler DNA that tends to have the base pairs repeat themselves over and over. They call them short tandem repeats. They use special enzymes that act as scissors that cut only in specific areas of DNA. Let's say we use an enzyme that specifically cuts a DNA strand in between the base pairs GAT-ATC. Whenever the enzyme recognizes the bases GAT-ATC in that order, it will make a snip in the DNA, making DNA fragments. Now remember, everyone's DNA is unique, so the fragments will be different lengths for each person. Scientists use a lab procedure called gel electrophoresis to then separate and measure these fragments for each person. The DNA that has been snipped with the enzyme into fragments along with a dye is placed in a charged gel agar-filled dish. The smaller fragments of DNA move faster down the gel. The larger fragments are slower and don't move as far down the gel. Here is a fluorescently dyed example of several samples of DNA that have just gone through gel electrophoresis to separate the fragments. A print of the results might look like this and be compared. On the left is the results of gel electrophoresis on a sample of cells found at a crime scene. To the right are samples of cells taken from the suspects. Whose DNA fragments match up with the results from the crime scene? If you said suspect number 2, you are right. That places that person at the crime scene. A forensic scientist can now report this as evidence to the police, a criminal investigator, and possibly to the court of law.